what I'm going to share and test on you for this DNA-based diagnostics. So you have learned the genomic, right? Proteomics. Proteomics already, right? Sorry, Leo, where are you from before? Undergraduates?
I'm from Unimas Sarawak.
Unimas, Sarawak. So,
Yep.
okay. Can you introduce yourself first before I start?
Oh, can, no problem. So, hello, my name is Leo. My true name is Yap Jijian. So, I'm 25, going to be 25. Oh my God, I feel so old. Previously, I do my undergrad on Unimas Sarawak, and I took biotech. I did biotech. So, yeah, I'm from KL. Okay.
Okay, 25 is not old. You have to put your age is upside down a bit.
Now
And
I'm comparing with the people living in the same house.
okay, next. Thank you, Leo. Just
Yeah,
a
sure,
little
no
bit so
problem.
that I know you. Sorry, this year I think we have been like this on the first during your first session with us before, right? That was when? October? Was it October, right? October.
October 11.
Sorry?
So the term was with Dr. Vanu.
Sorry?
You mean this course, DFM 522?
First, yeah, TFM actually started last month, right?
Yeah,
For
we
this
did we did
5
our first session with Dr. Vanu.
Yeah, okay. Okay. I will be in Penang end of this month, probably 20 something. So, so sorry, Penang, I think this year, last month also, there's a problem with water. It's beyond our control and it's going to be another one, right? On the 10th to 14, but I just received the email from the HR office says that we probably have to do have to sort of prolong the until 19, I guess. We'll disclose to you soon. Okay. Next, please.
Hello everyone, I'm Sana. I'm from Iraq and I did my undergraduate in medical microbiology in my country.
Okay.
Yeah.
Thank you. Next. Sana. Okay. Just want to know how to pronounce her name. Then that's why. Next. We want to start the class. Please.
Hi, I'm Kavita.
Okay.
Hi, Doctor. I'm Kavita. Yeah, I'm a part-time student.
Oh, okay.
Currently in Penang GH. My undergrad I did in UKM for microbiology.
Oh, okay. Bangi, right?
Yeah, Bangi.
Yeah, I was there years ago, but not microbiology. I'm from genetic background. Okay, next.
Hi, Dr. Atia. I'm another part-time student. My name is Yao. Y-O-W Yao.
Where are you?
Here.
Oh, okay.
Okay, I'm an undergrad from the Utah for biochemistry. Then currently I'm working in the Lo Kuan Lai Specialty Center, a private hospital in Penang.
Okay. Next. So both of you are part-time student. Next.
Hello, doctor. I'm Yi-En.
Oh, okay.
I'm from Utah as well, biomedical science background.
So same class with?
No, we are a very different batch.
Okay. Thank you. Next, you are also part-time student, no? Yen.
Oh no, I'm full time.
Okay. Next. Puspa.
Hello, doctor.
Hi.
I'm from Nepal.
So,
Background is medical microbiology.
from?
Nepal?
Oh, okay. Next. Ra'at. Is it? I don't know whether my pronunciation, is it my pronunciation?
Sadi from Iraq, undergraduate medical technician.
Okay, thank you. Who else? Oh, Leo already.
Hello, I'm Gao. I'm
Gao.
from China. My first degree is medical examination.
Okay. Thank
Thank
you.
you.
Song.
Hello, I'm Song Dai. I come from China. My undergraduate major is pharmacy.
Your undergraduate is what?
Pharmacy.
Biotech.
Pharmacy, no. Pharmacy.
Sorry.
Sure.
Pharmacy.
Pharmacy. Okay. Sorry. Thank you, Sultana.
Yeah, hi doctor. I'm Sultana. I'm from India. I'm also from pharmacy background.
Thank you. Who else? Everybody okay. Ra'at, you open too. That's why it's like echo. Okay, thank you so much. Most of the time I'm here, but I'll see whether once a month. Normally I go to Penang once a month. Sometimes twice, sometimes 3 times. It depends on meetings and whatnot. So I'll be there. Just catch me every. I think I don't have time to see you guys. Right. In on face to face. So anyway, I'll start this. How to share this? I think you can actually look at the slides. It's actually probably the same one with the one that Dr. Nurul Hasana already put in the ELN. Actually look at that. Share. Whether this PowerPoint is the same PowerPoint. I've got two PowerPoint here. Okay. So you can see only this one PowerPoint, right? DNA-based diagnostics. Yes or no? Hello? Yes.
Yeah, yes.
One. Okay. No, because I open so many. I started my meeting from 10 a.m. non-stop. Couldn't find time to go back. I'm still in the office now because yeah. So anyway, today we will actually share. I will share on the topic of DNA-based diagnostics. Basically, diagnostics, we can have protein, we can have RNA, we can have DNA, and for these diagnostics, what is the important pillar of healthcare system, which is diagnostics? Diagnostics, we need to basically get the right information at the right time to the right people. So that's how the diagnostics that we create or we buy or we get from somewhere, which should be relevant to the people and should actually use at the right time. Okay, so it's something like when you have symptoms of COVID, then what you need to do is you need to test at that time straight away, not until your symptoms are over, and you've got to use that for COVID, not for other diseases. Okay, so the guideline of selection of diagnostics, as we can see in these slides, we have to basically define test purpose. Is it for diagnosis, disease monitoring, or and you have to look at whether it's quantitative or qualitative. For example, if qualitative is your RTK COVID and quantitative is your PCR, whereby you can see the CT value of that PCR, real-time PCR. Okay, and we also need to review the market, identify the product available, details of the test available, and requirement for the training. Because for each diagnostics, we need to train. There are shit called, when we learn the ISO 13485 for the diagnostics medical device, which is relevant to human diseases, then we have the they call it the information for the user itself. Okay, so you need to basically see the details of the test itself. Okay, so whether it is required for training or not, we need to see on the what you call instruction on the box. If it is RTK, you just read that procedures, and you can do it on your own because it's self-testing. But if you want to do PCR, there's of course there's the process or procedures that you need training. Okay, a certain regulatory approval, have to look at whether the manufacturer meets the ISO 13485, CE mark, or FDA, or Medical Device Authority Malaysia. And also need to determine the test diagnostics accuracy under ideal condition. How we do that is by the test performance. So guys, I think if you do work in the private sectors, you need to order the diagnostic tests. You've got to do the test performance first, evaluate first, then only we see diagnostic accuracy, sensitivity, specificity. When it is good, then only you can actually use that. Okay. Another one is the determine the test diagnostics accuracy in clinical practice. This one is the test performance at end user level. Okay. And last but not least, monitor the test performance in routine use. So we've got to actually look at the quality control, proficiency testing, and we also need to train the end user. Okay. What is diagnostic? Why we need diagnostics? Diagnostics, it helps clinician to detect disease, determine the causes, monitoring patient progress, and improve outcomes. So we have different types of diagnostics, like diagnostics for detection, diagnostics for progress, like treatment. When you do treatment, then you need to see the performance, the treatment efficacy, whether it's okay or not. Then it's called monitoring. It's under the companion diagnostics, and you see the improve of the outcome itself. Okay, products include devices to assist in clinical chemistry and immunoassay, urinalysis, point of care testing, patient self-testing, and laboratory automation. So there are different types of diagnostics that involve with DNA as well, because it can combine DNA with other other complement for DNA, and we can also immunoassay we can also use for PCR, ELISA, something like that. So it's include for the use of diagnostics. You need to choose which one is the best, okay, and whether it's DNA or protein or RNA. Okay. So the role of diagnostics in clinical decision making, as you can see here, this is the incubation before the disease express the symptom. This is where you can get the symptoms, and of course, after symptoms, you need to treat the patient. So the treatment wise as well has role in this, as I mentioned earlier. So the incubation is actually screening, determine the risk of the disease. You need to see the polymorphism of what gene or what or to basically make sure that you know that this somebody has risk. So it can be in the incubation for the infectious diseases. It can be the genetic testing to actually look at whether you have high risk. When you have the certain polymorphism, you have high risk to get the disease if the risk is there. Okay. So symptoms, for example, rule out infections. Example, malaria, cholera, dengue, okay, and so on. And treatment is on the determine drug susceptibility. This is example, and initiate treatment, treatment monitoring, and test of cure. So these are the roles of diagnostics in clinical decision making, in incubation period, in symptoms period, as well as the treatment. Okay. This is the example of genetic testing, where you look at the changes in DNA and provide the diagnosis of genetic conditions such as Project X, and you also get information about your risk to develop cancer. So everybody know Angelina Jolie that actually have tests. So after that, of course, she went for the surgery, right? That's the test that you actually look at to know when you see the risk. Okay. So single gene testing, known genetic mutation in the family. These are the muscular dystrophy and sickle cell disease. Panel testing is related with high risk of cancer. This is just example, breast cancer or colorectal. And large-scale genetic or genomic testing, exome sequencing, where you look at all genes in DNA, the whole exome, or just genes that are related to medical condition, or clinical exome, and genome sequencing, the largest genetic test, and look at all of the person's DNA, not just the genes. So there are the term for this, we call it precision medicine, right? So if you want to look at the individuals one and see how to basically make sure that everyone get who has the symptom, who has the symptom being treated. So we need to actually look at everyone's detail, not as a group of people. Okay. So this is the global care testing of infectious disease market. This is 2019. So by infection type, this is the one, HIV, Clostridium. So Difficile and HBV, pneumonia, Streptococcus, and so on. So at the moment now, you will see that so many viral diseases that actually came to the picture now, right? Of course, we do still have TB, MRSA, SSB, and so on. Okay, so here you will see that when talk about the point of care testing for infectious diseases, it's actually increased from 2015 until 2025 because there are so many diseases that actually happen. And the worst scenario is where you saw the I mean, we all know about the COVID pandemic. So it's whether it's epidemic or pandemic. Pandemic is actually cause the whole whole country, all the humans. I mean, not to mention where are you from. It's actually from Asia to Europe to all the countries. Okay, so these are the global point of care testing for infectious disease market. Why I put this is because you are here to basically see beyond you as a student, because when you study all these diagnostics for the protein based, that DNA based, RNA based, the aim now is actually to probably create diagnostics. So you need to actually look at what is the point of care testing in the market. So as you can see here, these are some of the this is the infection type overview, 2015 and 25. So some of you from the private sector, so you've got to basically see what is the trend of the disease, and for you to produce probably, or to manufacture, or to get information of the diagnostics or point of care testing, so that you can actually do the evaluation before you can start using the test. Okay, so majority of the diagnostic tests used worldwide are based on the versatile lateral flow immunoassay platform, which is your RTK antigen. Of course, this is easy to use, but the sensitive device we've got to actually look at as well, because it's not that sensitive as the PCR or molecular method. Okay. However, there are many emerging technologies that are expected to improve the performance, accessibility, and adoption among the users in the global market. One of them is being microfluidic technology. We have sensor technology, imaging by microfluidic devices, are integrated point of care system for end-to-end fluid handling, processing, and signal generation. So this is all, this one is qualitative, but the microfluidic goes to signal generation is the quantitative. This is using sensors. So technology provides low-cost diagnostic tools to be used in home care or remote setting. Microfluidics or on-chip also have similarities to standard ELISA. Molecular diagnostics, lateral flow immunoassay platforms in terms of operation. Oh my God. Yet the use of microfluidic assays reduce assay complexity and enables multiplex testing, high throughput. When you look into the diagnostics, one is for user friendly and cheap. so that's why people are using the RTK a lot, where you can do self testing, but not all disease that you can do self testing. Well, before it started for pregnancy test, and now we can see many diseases that have the lateral flow immunoassay technique for diagnostics. And when we move to digitalization, then microfluidic device, chip, biosensor came in. So, but not many in the market yet, because I think for lateral flow assay, some of you, did you learn already the lateral flow immunoassay from Dr. Shahida? Yes or no? Not yet. Hello?
We are not yet lah. Part-time students are not yet.
Not yet. Oh, okay. So never mind. You will do next. I don't know when. Probably this middle or end of this month. Cannot be end, right? Because you have probably before 20 something, right? 20th. So that one, the lateral flow immunoassay or lateral flow assay, the production is a bit easier, even though production means manufacturing, but the development itself, it takes some time as well. But the as long as you have the proper machine placed, and the most important part for the lateral flow immunoassay is the humidity, because the membrane that you put on, that we use in the lateral flow assay strips, that you see line, when positive for COVID 2 lines, negative 1 line, that one is that part is very crucial, because the membrane cannot tolerate with high humidity. So when we produce, it has to be in the humidity of 10 to 20. Okay, that's low. If you have time to visit, I will see how. Visit Kelantan. In fact, in Penang you have, but not the proper one. In here in Kelantan we have. You will see the small one, but then the humidity is actually 10 to 20. So it will give like the shelf life of the strips longer. Okay. In other tests where we need the quantitative, I know PCR, quantitative means qPCR, but nowadays we basically the way forward that people are actually looking and developing is actually microfluidic devices, and also the chip that connect to a sensor. So it's this sensor, and of course this is the quantitative. Okay. You can see say on-chip immunoassay is are particularly promising, as it efficiently miniaturize and integrate several steps, such as cell lysis or extraction, purification, and amplification of the nucleic acid, and also detection reaction. So if you have lab on a chip, you will have all those straight away: extraction, purify, amplify, and detection straight away. But some actually have some, like probably the amplification and also the detection, and some has just detection, where you can get digitalized results at the end of the day. Okay. So ongoing effort to develop lab on a chip diagnostic tools include discovery of new biomarkers and develop integrated immunoassay design and materials. However, researchers are required to have equal focus on containing per test cost. Okay. So what does it mean when you want to create or develop diagnostic test, what you need to do or develop lab on a chip diagnostic tool, what we need to have is biomarkers, the integrated immunoassay design and materials, and of course we need to actually look at the test, the cost as well. So how costs actually reflect all these things, because if it is cheap then more people will buy, if expensive and not many people will buy. So even though it's quantitative, as long as you can actually detect the results, even though people actually don't actually look at how sensitive is the diagnostic. Okay. So they're talking about HIV generates the largest adoption and revenue. Now we have the COVID-19 and influenza, and of course the multiple drug resistant tuberculosis, because tuberculosis is still a major issues. Okay. Okay, this is the global point of care test. You can see North America here, Europe, Asia Pacific, Latin America, and Middle East and Africa. Okay, DNA-based diagnostics, what is that? Is explain the molecular science and technology used in a common set of diagnostic tools, including various types of PCR. Everybody knows the various type PCR. We have singleplex PCR, we have multiplex PCR, we have inverse PCR, we have assembly PCR. What else? Nested PCR. You all familiar with all those, right? Yes or no? Hello?
Not for all the variation, but the basic,
Basic
the
like
standard PCR.
standard PCR. Oh, okay. Because I teach second year undergraduate School of Health Sciences. That was in the syllabus. That is in the syllabus where I got lectures for them. So it depends on the institution, right? But no worries for your one, as long as you know the basic one for PCR, then you can answer your question in your exam. That's the most important part, right? For you guys. SNP analysis, single nucleotide polymorphism chips, next generation sequencing, and also microRNA techniques. So these are all involved with the. Okay, so we have polymerase chain reaction that use only the primers. We have probes, and we need to know how the PCR works, from the denaturation to annealing to extension. Are you familiar with PCR or not? I mean, you can tell like without any slide.
Yes.
Yes. Good. How about others? Yes or no?
Yes.
Doctor, we have like conducted the qualitative PCR with Dr. Venio, so we have as a background and also like in practical too.
Yeah, in practical before this or now?
Yeah, now like we had it.
Yeah. We will have.
Dr. Venio.
You already have with Dr. Venu?
Yeah, qualitative PCR. We did it. Sorry, quantitative one.
Quantitative means qPCR.
Yeah, qPCR.
Real-time PCR, right?
Yeah.
Oh, okay. Well, that's good. So you will learn that for practical. In this one is the normal PCR. So you learn the most difficult part first. Then should be okay for this one. The only thing that I'll try to trick whether you know the whole detail of that or not. Okay. microarray technology that explains SNP's chip technology and discuss the use of SNP single nucleotide polymorphism chips in diagnostic application. We have next generation sequencing technology where it looks at the various next generation sequencing tools and discuss the important diagnostics application that this machine can offer. In Penang, the place that you can get or see real time, next generation sequencing is called, there's Prof. Alex Institute. Wait a minute, can't remember what is the there's one that actually have the analysis and whatnot. Okay, micro, can't remember it. Alex. There's one ABRC. There's another one. Okay, I'll tell later. In over here in Health Campus, there are three units of next generation sequencing in School of Health Sciences, as well as the medical sciences. The microdiagnostic technology summarize how microRNA works and its application in diagnostic development. So this is the biosensor, whereby this is just example for you to actually look at the Hinge DNA origami biosensor that designed to detect the response of the SARS-CoV-2 virus that actually involved with the sequence of the COVID. This is RNA because COVID is RNA virus. So we tend to overlap a bit. So, okay. If you want to look at the diagnostic techniques for COVID-19 and new development, please look at this paper. I can send you if you want to read more on this. It's actually explain on various diagnostic techniques for COVID-19. Okay, and this is the real-time the polymerase chain reaction, which is PCR, that basically utilize the dry reagent base. So instead of we have the PCR with the liquid reagent, so what we did was basically dry the reagent with the enzyme stabilizer. So the reagents include everything. The reagents, buffer, plus the DNTPs, magnesium chloride, enzyme, tag polymerase, and we actually add the enzyme stabilizer and dry. So when it is dry in the PCR tube, so the users can actually just put samples and then make sure it's homogenized, then put straight away in the PCR machine or thermocycler. Then you can actually run gel or gel. That's the common one. You can have other technique, which is lateral flow assay, also okay, or sensor, or that's the detection part. So this one is basically this was published when I was doing PhD, and this one is actually been done by my student, which we actually combine the PCR for DNA amplification and with the lateral flow assay. So after running the PCR, the amplicon or PCR product is actually was placed on the lateral flow strips, and you can see bands, positive and negative. I think I'll show you later. So this is and we actually move into the sensor. So the detection part is in the electrochemical sensor. PCR is the in vitro diagnostics, common one for in vitro because what we and the temperature is done in 3 temperatures, right? It means you have to have denaturation, you have to have annealing and extension in 3 different temperatures. Okay. There's another method called isothermal amplification, and LAMP is one of them. So because of the PCR, because of the PCR, we want to make sure that we can actually use amplify the gene without having the PCR machine. So you can use heating block or water bath. So then what we can actually utilize is our LAMP or HDA or CPA, and this is another one, CRISPR, where if we want to actually increase the sensitivity, reduce the limit of detection, increase the sensitivity, then CRISPR is being used by some of the researchers. So they basically use CRISPR-Cas12 and 13. Okay, so this is to show you that the illustration of that, you need to have the amplification and reagents. So this one is RT, means from RNA to DNA, then amplification happen for the DNA, and then we continue by CRISPR, and after that we can actually read by lateral flow assay or the turbidimeter or fluorescent machine. Okay, and this is to show you. Wait a minute. So this one is one pot reaction for detection SARS-CoV-2, CRISPR-12 reagent with RT, LAMP, master mix, and you've got the amplicon, and you can actually read the signal. Okay. So basically, when you have the samples, you need to actually extract RNA of this, and the RNA is actually converted into DNA. Then this one, Cas12, will basically use to ensure the sensitivity is high, and you can read with fluorescent reader or literature. So this is the CRISPR-12. But for CRISPR-13, from the samples, RNA extraction to you have the RNA amplification, and from cDNA to double-stranded DNA, and you've got to transform into RNA amplicon again and use CRISPR-Cas13 and can read to the fluorescent or electrophoreal strips. Okay, so this is example, the multiplex PCR. So the multiplex PCR can actually amplify different types of gene. Okay, this is VI. Multiplex PCR VI is one of the protein on the capsule of the Salmonella. So multiplex PCR, there's another one that can be utilized for the multiple targets. Then you can actually detect the presence of different genes from the same organism or different proteins from different organisms. So that's the beauty of multiplex PCR. Okay, so as you see here, the molecular diagnostic market, you will see that it's actually increased from 2017 to 2025. This is the forecast. So what you need to actually look at is, you see, this is huge one, is the infectious diseases, and you have the blood screening, oncology, histology, and others. Okay. Why I put this? Because always the tropical regions that you can actually have more diseases, especially for the infectious diseases. So this is the 10 global hotspots of neglected tropical diseases. So if you guys want to create or develop the diagnostic test or packaging, rebrand and send and sell to the others, you can actually look at the hotspot that's of this, and these are the incidents of selected of the incidents for tuberculosis, HIV, diarrhea. So that's how you can see for this one, this is good for you to have the test for diagnosis. So one of the disease that have higher incident rate is tuberculosis, where you will see in this region. And where are we here? So it's everywhere actually. So that's even though tuberculosis is the test is being developed everywhere, you have so many tests in the market, but we still need to make sure that test is simple, easy to use, and also affordable. Okay, we have malaria at this region, HIV, that's the Zika, cholera here. Okay, so this hazard health by. Okay, so trends in causes of death, you will see there's this respiratory, diarrhea, HIV, TB, and so on, and this is infectious one. Okay, so the characteristic of the diagnostic test you will see that Ashworth criteria by WHO, where if you want to have the if you want to develop the test, it has to be affordable, sensitive, specific, user-friendly, means that it's simple to perform with few steps, and if possible, we don't need to train, or you can actually just read on the box or on the pamphlet, or with minimal training. It's robust and rapid. That's results available in less than 30 minutes. Equipment free, and we can deliver to those who need them. Okay. If you can actually look at this is the evaluation diagnostic method. We started with microscopy, where you can see the morphology of the organism itself. It's followed by gram stain, the culture test, check for the antimicrobial susceptibility, and also look at the acute and convalescent antibody, molecular antibodies, antigen detection, real-time polymerase chain reaction, microorganism detection, as well as drug-resistant genes, and mass spectroscopy. So you will see that this is the time of the identification. Meaning is weeks, hours. So people are looking for the short period of detection, but cheap, of course. But you will see that in if culture, it depends on the bacteria or organism itself, whether it's fast growth or slow growth, or in medium. So you cannot just, okay, you put so many nutrients inside there, you think that it can grow fast. No, it depends on the duplication time of the bacteria, if it is bacteria, and how the growth cycle of each bacteria itself. Okay, so have you seen this, where we are doing in the lab? So this PCR is in lab on a drone for PCR, enable with convective flow thermal cycling. So probably it's not, yeah, this is in US. Okay. For diagnostics and microbiology, you can divide into three, which is one is conventional diagnostics, molecular diagnostics, and rapid point of care diagnostics. All these three are complement to each other. That's what I see. Because let's say for conventional diagnostic, you need to basically do the radiology, X-ray, and biochemistry, hematology, and pathology, and microbiology. Then you can get, this is approximately 1 hour, 12 hours or more, and 24 hours. So these are the things that you need to actually look at. So molecular diagnostics, normally for human genetics or genetic diseases, or molecular microbiology lab. So look at the rapid or multi-parametric test or post-culture molecular identification. And this is human genetics and pharmacogenetics, is tumor typing, drug metabolism, and etc. And this will basically have approximately 1 hour to 12 hours or more. But rapid or point of care diagnostics, you can have approximately 1 hour. Even if you can actually reduce the test period, the time for us to get the results, then it will be better, as this will have the immediate healthcare decision whether what type of drugs that the doctors can actually prescribe, antimicrobial resistant, antimicrobial antibiotics, and we have to look at which genes resistant, which drug is resistant. So for molecular diagnostics, can be used for microbe identification and drug susceptibility. For immunodiagnostics or glucose testing, you just poke your hand and actually see the results. Okay, and this one is microscopy based. So that's the diagnostic microbiology, but what we want is actually less testing time and accurate. Okay. So nucleic acid amplification technology, the simple one is we need to have three steps, which is sample prep. You cannot run away from this. Sample prep, amplification, and detection. So sample prep, you can actually use boiling method, commercial kit, automated DNA prep. Can have the amplification via PCR or isothermal amplification, such as HDA, helicase dependent amplification, LAMP, loop amplification, RCA, rolling circle, TMA, CPA, and NASBA. And detection can be done via agarose gel interferosis, by sensor, dipstick, and PrEP, and others. We have ELISA, we have others as well. Okay. So point of care nucleic acid platform, you can actually look at this. This is PCR based and this is isothermal based. You can actually look at the platform in terms of the sample prep, whether it's included or you need to have separate sample prep. So for this YY is actually the sample prep is inside there already. So you just put in the samples, you get results out. But for this one, you need to prepare the samples and only amplify. Okay. And detection method, you can have real-time fluorescent or real-time bioluminescence or real-time turbidity. Okay, this is nucleic acid electroflow assay. Electroflow. Okay, so you can actually look at the time of the each platform. Whether it's acceptable, you want to have the shorter time, then you look for other method or platform. If you want to go for it depends on your requirement. If you can tolerate with longer, a bit longer, and you get good results, then you can buy that. Okay. So in terms of the current isothermal nucleic acid amplification method, we have this is RNA, this is for DNA, HDA, RPA, and we have the LAMP, CPA, SMART, AMP, RCA, RAM, SDA, NEAR, NEMA, ICA, XPAR, BAD amplification, PG, RCA. So the one that you will learn, you will actually listen to everything, but please focus on the isothermal, the PCR, and also the isothermal that you need to focus is HDA. Helicase dependent amplification, because this is so many. So I don't want you to get crazy. So just focus on the helicase dependent amplification and polymerase generation. You know why? Because the rest is yeah. How do I put this? The rest it's a bit complicated. I will show you later, but for HDA the reaction is sort of similar with PCR, but the isothermals means one temperature, which is 65 degrees, and the amplification happen via enzymes. Okay. So why okay, the amplification is basically from the DNA replication in vivo. So this is how the isothermal amplification been created, because let's say, for example, here for PCR we used to unwind the double-stranded into DNA into single-stranded via temperature with 94, but this one we use helicase. And we also have the single-stranded binding protein. This is single-stranded binding protein attached to unwound strand, preventing them from winding back together. So that's the beauty of this, but you need to add this DNA binding protein. And you have DNA polymerase for the catalyze of elongation of the leading and lagging strand. Okay, you are familiar, I guess, with the DNA primase Okazaki fragment and the DNA ligase. With this is the lagging template. So the leading template, where it goes straight from this, and then when you have a DNA polymerase, you will actually synthesis new strand. Okay. So okay. Now this is the polymerase chain reaction. What is this? This an in vitro technique for the amplification of a region of DNA which lies between two regions of unknown sequence. So the PCR amplification is achieved by using oligonucleotide primers, and what you need to actually know is what the criteria of PCR. The primers, which is short fragment of 18 to 30 mers, single-stranded oligonucleotide, and is complementary to the outer regions of the known sequence. Okay. So what are components? Components, you can have like primers, DNTPs, enzymes. It is like this. It's just after that, when you apply the 90 degrees, it will separate the DNA as well. And when it goes to the annealing temperature, which is 2 to 5 degrees from the melting temperature of the primers, it will bind, the primer will bind to the template, which is annealing process, and the extension happen at 72 degrees. Anybody knows why it's 72 degrees? Yeah. Why is 72 degrees? Leo, you seems to know well about PCR. Why is 72 degrees? Yeah.
I came across this before. Wait, let me think. Why 72 degrees?
It's because of what? Because of the
I think it's because the
Polymerase.
that was the best for the polymerase. Yeah,
Ah, yeah.
PEG polymerase.
Yeah. So remember this 72 is because of the optimal condition or reaction for the TAT polymerase. This one, of course, you need to actually look at your primers, and this one is for the denaturation of the double-stranded into single-stranded. Please remember this figure. Okay. Okay. For isotomal, you will have so many here. Yeah, what you have to focus is this one, the helicase dependent amplification, whereby it's similar with the PCR, because you need to have one pair of primers. The rest, like LAM, you need like four primers. Sometimes when I produce the COVID LAM, it's actually 6 primers. So is it this 6? Yeah, 4 primers, 2 inner, 2 outer. Okay. So this one, what you need to actually look at is actually is unwinding happen when the helicase is there, and you've got to have the single-stranded binding protein to avoid the self-annealing, and amplification happen with BST polymerase at 65 degrees. Okay, the PCR is 72 degrees for the PCR. PCR require 3 temperatures, but this one require 1 temperature because of the BST polymerase that works well. Optimal condition for the BST polymerase at 60 to 65 degrees. Okay, so the rest you can actually look at that. This is another, just to show you that there are types of isotomal amplification. I've done this with my student. I've done this as well. This one as well, and this one. But yeah, no worries. You just know PCR and the helicase. Okay, so this figure, please remember so that you know where the double-stranded and helicase unwind, the DNA duplex. Oh my God. In the presence of single-stranded binding and necessary protein to avoid self-annealing, primers to anneal to single-stranded DNA polymerase, which is BST polymerase, extend the primers, and amplification happen. Okay? Is it simple?
Yes, not so hard.
Yeah. So hopefully everything goes well with your exam later. Okay. So this is new of potential of new technologies to address unmet clinical needs. You need to actually look. Unmet need is rapid testing of clinical specimen with less than 60 minutes. Rapid testing of clinical isolate less than 60-minute, point of care, near patient testing, simplicity, syndromic testing, screening of infection, resource constraint setting, infection control, hospital epidemiology, and discovery of emerging pathogens. So these are the examples actually involved, and the potential technologies are, as you can see here, single-step molecular cartridge-based test, MALDI-TOF, MS. This is single-step molecular cartridge-based test, LAM. This is also single-step molecular cartridge-based, highly multiplex single-step molecular cartridge-based. These are coupled to magnetic resonance, biosensor, biomarkers. This is similar with the on the top one. Next generation sequencing, PCR with ACyTOF, and next generation sequencing and PCR can also be coupled with the sensor. So when we develop diagnostic tests, this is determinants of diagnostic test design for developing countries. So we've got to actually look cater for the clinical knowledge, disease and pathogen specific information, the available biomarkers by sample type, patient characteristics and availability of specimen banks for test development and validation. And second is technical specification. We've got to cater for sensitivity and specificity, throughput, whether it's high throughput or low, shelf life, is it 3 months or 1 year or more than 2 years? How about cost? Is it portable or not? And how to do the waste generation? What are you going to do with that? You can actually look at the when you test the COVID-19, which is RTK antigen. What is the waste there? You throw away, right? So what is that? It's plastic, right? So you can actually look at the dig all the information there. If at the moment now, I think everybody is using that around the world. So you just imagine how plastic can be, because we are not using the biodegradable, degradable plastic. So you will see that the waste from that. Okay. So you've got to cater for the available infrastructure, whether got electricity or not, water, IT, telecommunication, maintenance, healthcare provider training, product distribution, and other instrumentation, geographic factors, which is prevalence and incidence, species and strain variability, culture factors that encourage or discourage testing, and also availability of therapeutics, a regulatory requirement, with the barriers to entry, exclusion to substandard competitors, requirement for in-country studies, and external agency evaluation. And other consideration is the financial constraints, the sustainability of business model for manufacturer, distributor, and healthcare provider. Okay. So unmet diagnostic needs in clinical testing. This is for outpatient clinic, emergency department, hospital and ICU, pressure control and hospital epidemiology, public health surveillance, biodefense and imaging infectious diseases, resource constraint setting, as well as special patient population. So these are the unmet diagnostic needs for clinical testing. What's the time now? You're supposed to have tomorrow, today. I just talked to Leo before I give the 7:20. You're supposed to have practical today and lecture tomorrow, right? Right? Before.
Yes.
Yeah, only today that we said that okay, because practical is not there. So I would like to stop here, and then can I proceed tomorrow from 6 to 8?
10, doctor.
10. Okay. So at the end, I will basically, because the PCR, the practical is PCR. Okay. So whenever COIS is now suffered, is suffering for dengue. Is it dengue? Yeah. So when he's back, then you can have that, but I will discuss with you guys the results of the PCR for last year. So now I think it's easier that because last year's one, last batch, the PCR is some successful, some not. So that's the good one for you to learn. But when COIS is okay, you can actually do your experiment. You can have the results. So the reason is I'm not sure how many days that COVID that COIS is okay. So is it possible? Now I leave it to you to decide. It's not me to decide. No, because you have another practical, right? Shahida got one practical, right? Dr. Shahida? Hello?
Yeah, I think so.
Yeah, I think you have, I'm not sure whether it's one or two. So if I discuss that, you can have the experience to do PCR and see your report, your results. But I don't know, I would like to suggest that you use the results from last year's one and then do the report, then it will be earlier rather than you cram together with 2 or 3 reports. I'll leave it to you. If you say yes, then I'll of course I will discuss with you straight away. But for the report itself, you want to wait for your results, for your PCR result after the your practical class, then you have to wait until it's done. But if you would want to write the report based on the result from the previous one that I already have, with some okay, some not okay, so you can discuss it, why probably this happened, probably that happened, or something like that. Which one do you want? Either way. Leo said either way is okay. So you will be crammed with your. I'm okay with that. I can just discuss and then when you do PCR. Later you can use your results and then do the report. Yeah. Yeah. Because I can give the results. The results, then you can just write the report. Anyway, for your practical to do PCR, then you can do with COIS. But the result is okay or not okay is actually depends on your experiment. It will be the same one, I guess, because I'm not sure whether if your pipetting is good, then you will get all okay, and there's no other explanation why it is not okay. Right. Okay, I want to know from all of you. You want to after tomorrow, then you can write report based on the previous one. Yeah. Yes, Yen. Yes, Leo already asked me just now. Then I stop. Okay, I'll stop here.
Doctor, if
Yeah.
we are to write report based on last year's result, then we will have no practical class.
No, you will have. You will have practical class, the experience for you to do PCR, and you can see also your results later. Yeah, wait a minute. My brain cannot talk. You can talk now. Writing report based on last year's results. How about agarose diaptophrosis? No, after the results that I will give you is the results of the agarose gel autopharesis results already. The picture of the gel. Do I have them tomorrow? The result is basically, when I say result, it means that the agarose deadline of the report. You have to ask Dr. Nurul, because you have another report. So if COIS is, I'm not sure, probably it takes few more days. So if you can actually, what's the time change in this case? Yeah, timeline. Yeah. Hello, you can talk to me. Why so shy?
I mean, for me, either way is fine lah. But doctor, can you decide a deadline for us?
Deadline, I need to ask Dr. Nurul, Dr. Nurul, because you have another deadline for,
Preferably on 20 something lah, because there's too many assignments currently.
Yeah, that's why I said, if I give now, with that one, then you can write straight away.
Yeah.
Because I think it's like write straight away, and then you have time for another practical to write for that practical.
Yep.
Correct.
So I think for me it's okay to use the last year result to write.
Yeah, it's just that you can have your practical, normal practical for you.
Yeah.
So you will have all the reagents, and you will do the pipetting, put in the PCR tubes, and you will run the PCR, and after that you will run the agarose gel as well.
Yeah.
Yeah. But it will take few days for you to, as long as they no practical. It will be a practical. No worries. No worries. In fact,
I think okay lah. We can stick with the last year result lor. We can straight away start to write already.
If not or I will tell you tomorrow. I'll ask Dr. Nurul when is the deadline for the.
Yeah, so we can use the practical result last year to write, and then let us know when is the
yeah.
the latest deadline you can get for us.
I think because you will have the, is it on 15 or 20? Your workshop. Wait,
the workshop done already.
No, the work done. Which workshop are you talking about? How many workshop that you have?
You mean the science quest workshop that? Okay. Okay, understand. All right, there will be another workshop.
I think there will be another workshop, right?
Yes, it is.
Yeah, I guess the practical should be the practical report. You should submit probably before that. I think I will ask Dr. Nurul. We'll get back to you tomorrow. Okay?
Okay, sure.
Yeah. So yeah. If you guys, because last year's they all like to I would say curious about others, because I brought the PCR, because I developed PCR dipstick. PCR dipstick means the PCR, and after that you have to do PCR. And after PCR is the lateral flow assay. So I brought all those things. The PCR you know already how to do that. So we can have the PCR product. Then you can load on the strips. Then I will explain to you what happened behind that. You can have. Will you be here during the practical? I have to see. I will be there from 22nd and 23rd. Probably if you guys okay with 23rd afternoon, or yeah, I'll be there from 23rd until 24th or 25th January. Okay, it's
Afternoon can't, doctor, because working.
yeah. Sorry.
After work, we can make it, but in the afternoon, it'll be difficult for us to take leave.
Yeah, can. No worries. I just stay. I stay normally when I go to Penang. It's the what is the opposite inform is the hostel, right? Harapan. I just stay there. I'm not staying in the So it's okay with me. So we'll get back to you because I need to look at the details as well. But we put the KIV 23rd for if you want to do PCR on that particular time also okay, and I can bring the but if like that, then you just do PCR, then the results for the report should be from the previous results. Because 23rd is 23rd. That one is probably after your workshop. We cannot be like that, I guess. It should be before.
Presentation, actually.
Sorry?
The workshop presentation.
Yeah, I don't want to disturb that. I think that's the letters that you can submit your, I think before workshop. But if you do that and that time, because Kois will say that, I will practically ask them when you guys are free for him to actually show to you guys. He asked me whether it should be 6 to 9. I say yes, because some of you are working, right? Yeah. So, by the way, for those who are part-time, are you in Penang or in Ipoh or what?
Penang.
Penang, so okay lah. Is it island or that main land sebelah?
Island.
Island, so okay lah. Island is so small, right?
Jan.
Oh yeah, understand. So yeah, I think the best way is for Kois to have the practical for you earlier. If not, then we can do it. Just say okay, 23rd evening.
Okay, if that's the best we can do, then 23rd.
Yeah,
then.
if you want to, then I'll be there
Okay.
at the time, 23rd. Then we can do together all those, everything.
Yep, sure.
Yeah, but your practical, your report should be done earlier, as we will discuss tomorrow on the results and what are the things that you need to put on your report. Is that okay?
But wait, doctor, the 23rd is the.
What is?
Wait, it's not our workshop, meh? According to the calendar here.
Yeah,
Oh, you have workshop?
Oh yeah, 23rd is the. It say continue lah. I'm not sure whether we can finish on 22 or not. Actually, 22 and 23rd is we have workshop on that period.
So workshop is also 6 to 9.
Yep.
Oh.
We can do 24. 24 is free. We are free for the whole day.
24 is okay. I actually have Senate meeting in the morning, 24.
Mm-hmm.
Then evening should be fine.
Okay, then 24.
But I'll confirm by tomorrow. But
Okay.
as long as I know that 24 you guys are free.
All right, sure, no problem.
Okay. Thank you. Minutes too. Okay.
But if we can finish, then it should be okay. Ian say we can finish bro,
What? Sorry?
for the workshop.
Oh, workshop. I'll
But
ask,
to be safe, we place 24 lah. Let's say today anything on that day you cannot finish, then
oh, you afraid that you cannot finish the workshop on
just
the
afraid lah. I don't know. There's the reason why they put on 24 also, 23 also ma. So I'm not
Oh,
sure
okay, okay.
we
Then,
we don't have any authority to change the
yeah,
schedule
no,
everything.
no worries. It's like this. I think then it's better for me to discuss with you tomorrow, if like that. Tomorrow, the last year's result, and you do the report. And then whenever Kois is available, he will do the practical with you. But I can be like this. If you want to, I'm available. If you want to discuss anything, I will be there. I'll have to ask him because I'll fly to Hong Kong on just few days. Then after that, I'm around 20 days. Wait a minute. On the 6th from here to KL 7 to Hong Kong. Symposium on the 8th, 9 come back already. 10 morning I will reach here and then straight away in the office. So, but 10, we have a problem with water in Penang, right? Right. Yeah, so we'll ask, yeah, no water. Over here we have, last 2 weeks, we have water, but it's from flood now. Cannot use anyway, right? Yeah, so, yeah, yeah, I'm here. After that only I will go there on that 20th. So, but anyway, the practical won't be done. It cannot be done on that period. So it should be probably earlier. We'll ask Kois, and Norris will find the best alternative for you, best day for you to do the practical. Okay. Okay. Thank you. Bye, everyone. See you tomorrow. See you tomorrow. Actually, you can use the same. Oh, I'll have to do it again. But same one. Because this is my, when I do it, if then, never mind.
Okay.
So,
don't come, no water.
no worries. I won't go there.
I'm escaping to KL already tomorrow. Ha 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 ha.
Oh, okay. So then, because it's the practical should be done after that, right? Because you were not there anyway. Oh.
I don't know. I'll be back after water supply is back.
Yeah.
When water is here, I'm here. Water is not here, I'm not here.
Because the water, they put there 10 to 14. I just have to go back. It's 30 minutes. This is the Indo. Okay. 10 to 12. University, because previously university actually encouraged for staff to work from home on 10 to 12. 10 to 12 or 10 to 14. But now it's actually extend to also to 15 to 19.
I think extended to 19.
Yeah, extended to 19. What date is 19? What day? This is Friday.
19 is Friday. Friday.
So you have that week as well. That week is 22nd, 23rd, 25th. 25th, Thaipusam. Oh my God.
Yes.
Okay, you got holiday again. In Penang. So Thaipusam, we don't have holiday here. Not holiday here. On Penang, yes. I got Friday. Of course, you go back and then don't come back. Yeah. 26, right? You go back and then. Okay, never mind. We'll discuss. We'll tell you tomorrow. Okay. Thank you, guys. Bye.
Bye-bye.
Bye.
Thank you for the lecture today.